So incidentally, I'm not only closing the symposium, I'm closing a session that features two of former George Cody postdocs. George Cody is at Carnegie Institute in Washington. Oh, actually, and there is another one of his postdoc. Hi, Jim. So I arrived at ELSI exactly a year ago. I arrived just before the previous symposium, last year's symposium. And around that time, we started to get our band together, messy chemistry band together. Narisa Iskandar, if you're here, thank you for that image. So today, I'm going to be presenting the work that is done by all these wonderful members of the band. And this is not complete band. This is just the work I'm going to talk about. So what do we mean when we talk about messy chemistry? First of all, I am a chemist myself. So the way I was taught to approach chemistry is to take reagent-grade material, ideally the ones that just arrived from Sigma Aldrich and hasn't been sitting on the shelf for a long, long time. Mix them in a specific way and just try to maximize yield, transform them in a very particular way to another set of compounds. When we're talking about origin of life, though, we usually think not about single reaction, but things like HC and polymers, which are completely messy, complex systems, polymeric systems. We're, of course, thinking about Miller-Urey experiments. So in addition to a few amino acids, it's a very, very complex mixture of monomeric and polymeric materials. We might be thinking about tholins of Titan. So some believe that this brownish color of the picture that was taken by Cassini, Huygens, Lender is due to complex polymeric matter. Or alternatively, you can be thinking about insoluble organic matter that just Yoko told us about. When we think about our life, we think about all of those metabolic pathways, and when we talk about life-like processes, we're probably talking not about a single reaction, but some subset of this network. So this system, of course, very different from those. This system is under very tight control performed by enzymes. So what is messy? Well, this is a famous quote from Steve Banner. It's one of his origin of life paradoxes. He seems to think when molecules given energy and left to their own devices, they devolve into something that is better suited to pave roads, but not to start origin of life. With all due respect to Steve Banner, and I truly honestly mean it, we disagree here. So what is messy? We're, of course, taking cues from systems chemistry, and there are a couple of wonderful talks on the subject given earlier in the symposium. But systems chemistry, per se, can often mean a very defined, very small network. So what we're doing here, we're taking the systems chemistry to the next level. We're talking about chemistry of complex interacting components which are under very limited control. This system are not necessarily unstructured, it's just the structure of them is not apparent. And in our mind, origin of life is the transition from these messy chemistries to well-orchestrated biological system. So what we do at ELSI, we treat the messy chemistry network as a single entity, and using computer and experimental modeling, we try to decipher the structure of this entity, and we also poke at it to figure out if there are any emergent phenomena to be found. So first I want to talk about tangible messy chemistry. Something like HC and polymers are notoriously difficult to analyze. And as Martha Grover eloquently explained already, esterification reaction is a reaction between carboxylic acid and alcohol. This bond is somewhat reminiscent of a peptide bond, but it's easier to form. Orgel in 2003 somewhat reluctantly proposed that peptides can be potential ancestors So polyesters can be potential ancestors to peptide. And alpha hydroxy acid, that is alcohol analogs of amino acid, are shown to be polymerized by ribosome. And there is, of course, renewed interest in the context of origin of life with some beautiful work, well, if I say so myself, coming out of Center of Chemical Evolution in Georgia Tech. So what we did here, we messed up the beautiful clean system that Martha showed us earlier. So, and this work was led by Jim Cleaves and Kuhan Chandru. So what they did, they took 5 different alpha hydroxy acids, they subjected them to wet-dry cycles, and they got a whole bunch of products. And when you're thinking about it, if you take this mixture of 5 and you assume they're only going to make a 20mer, even in that case, you end up to the 5 to the 20th unique sequences. And the idea here was, here we are going to have a diversity generating synthesis that will create functional polymer that we can draw from upon need. So far, it's been tough to analyze this, and it's what Hongo-san and Nick did. They were able to decipher something like 43,000 unique sequences in mass spectra alone. So the next question is, can messy polymers be functional? And the answer is yes, in principle. And here is one old example coming from Sidney Fox. So what he did back in the 60s, he took mixtures of amino acid, he treated them. And he ended up with like those interesting looking structures that he called microspheres. So there are very mixed feeling about this work. So the good about this work, he actually in few different cases have been able to show that these microspheres are catalytic. The bad about this work is that catalytic activity is usually small, especially when compared to grandiose claims he made. And he never even tried to provide any mechanistic explanation to why these microspheres could be catalytic. And so the ugly about his work is he had some very unsubstantiated claim, for example, of non-random incorporation of amino acid. He did nothing to that effect. He claimed that he was making linear peptides, and it's actually probably not true because this polymerization and microsphere formation only seems to be working when a glutamic acid was added in excess. So what glutamic acid has 2 acid groups that both can participate in peptide bond and provide branching points. And most outrageously, toward the end of Fox's career, he claimed that his microspheres are lifelike and even conscious. Well, and then throughout this symposium, everybody found a paper to bash, and unfortunately, I'm bashing here somebody who's been dead for 20 years. I don't feel particularly great about it. So what Fox did in his time, so he organized a number of conferences in origins of life, and this is, I'm talking about one in 1965, and at that conference, he had a talk by a biophysicist from Stanford, Marston Blois. And he gave a very interesting talk talking about thermal or prebiotic melanin. So what he did, he took histidine, he treated it, got black tarry polymer, as you would expect. He realized that his polymer is not like biological melanin, which is more structured, but quite messy. But he saw it as an opportunity. He said, well, maybe it's a good thing. Maybe we can have a lot of different catalytic sites that are stereospecific, that can bind substrates and catalyze reactions specifically. So Blois never continued his research, so he made a career in melanoma research and bioinformatics. So that's the only place where his opinions appear. So can we actually build protoenzymes? And we probably can, but we need to approach this more systematically. So what is an enzyme? Enzyme is a molecule that has a catalytic site that actually participates in catalysis, and it's usually scaffolded by RNA or a peptide scaffold. And the function of this scaffold is to specifically bind, orientate substrates, and not only that, but provide environments that are different from surrounding water that might promote the reaction. So in synthetic chemistry world, this function of enzyme was quite successfully approximated by so-called dendrozymes. So dendrozymes are based on dendrimers. Dendrimers are like these fractal molecules, regular ones. So the way you synthesize them is you have your catalytic site in the middle, and you add upon it generations of branch polymers. And of course, this molecule is very engineered, so you can change the chemistry of each layer of each generation here to get very specific reactivity. That's not something we're looking for when we're talking about prebiotic chemistry. However, cousins of those dendrimers called Hyperbranch polymers retain a lot of their properties, of course, in less controlled way, and often they can be synthesized in one-pot synthesis and maybe some prebiotic conditions. So what I try to do here is actually have an efficient assay of catalytic enzyme-like activity of hyperbranch polyesters, polymers in this case. So I chose reaction and its camp elimination. And I hope you don't run away when I say this is a reaction-based catalyzed oxidative ring opening of benzoxazole. This reaction of no particular interest to prebiotic chemistry, what it is a reaction very sensitive to medium environment. So it proceeds quite sluggishly in water, but it proceeds quite fast in less polar solvents. So my thinking was here, if we build a catalyst that can provide microenvironment that are more hydrophobic, like let's say water, and I wanted to run this reaction in water, can I actually make this reaction go faster? So I started building a protoenzyme. So I synthesized hyperbranch polyesters here. So I use citric acid, which is multifunctional carboxylic acid, glycerol, pardon. I threw in triethanolamine. It's a base catalyzed reaction for the active site. I was able to prepare this polymer quite easily. So citric acid is quite polar, so I also had a system made with adipic acid and methylsuccinic, and these have hydrophobic moieties for compare. So, and this is one example of adipic acid. So this is my spectrum. And as you can see here, it is quite messy. There's a lot of different compounds in that mixture of polymers. But what I can tell you, they're all quite short. So we're talking at maximum 7 mers and 8 mers. And here are some results. So when you use monomeric triethanolamine, well, that's the rate of the reaction in red. When you use citric acid polymer, the reaction proceeds somewhat more fast. And actually there is like factor of 3 increase when you switch to methylsuccinic acid polymer. So what can we learn from here? Short branched oligomers can be efficient catalyst. And of course, efficient here, I want to take with a grain of salt because factor of 3 is nothing compared to what enzymes can do. But a few things to remember. Enzymes are hundreds of amino acids long. These guys here are only 7 and 8 mers. I know for a fact I have polymers in the mixture there, they don't have active site at all. And some of the active sites might be decorating the periphery of the molecules and not the inside where you want them. So a lot of biochemists are more comfortable when we're talking about peptide-based catalysts, so we probably can do that too. And we can do it through DEPSI peptides, something that Martha described earlier in the symposium. We can try some sort of a direct synthesis of branched peptides. Let's say we take lysine, and incidentally, branched lysines are quite well documented in different contexts in the literature. So in this next work, direct synthesis of peptides was done by Masashi here, Richin, and Kuhan. So what they did took a variant of Fox's synthesis. So they went and scorched solution of glycine for very short periods of time. So you can see the formation of dark tarry material. So then what they did is actually analyzed yields and lengths of those peptides. So what they're finding that you can make some short peptides when you're doing it that way, that would form in like first 20 seconds, and then they tend to degrade into this dark polymer. So is there a way to somewhat bias this reaction so we can only get peptides? And they decided it is conceivable that this can be done with periodic heating and cooling of this solution. And this is incidentally a regime that is known in natural hot springs, geysers with intermittent boils. So they built this beautiful apparatus. So this apparatus can deliver flashes of heat, and it can be immersed in iced water for quick cooling. And here is another one in action. Well, sorry about that, but you could enjoy those intermittent boils here. Well, and that just took actually a lot of work. The guys tinkered a lot with the type of heating block, with the concentration of glycine in this case, with the pH, and like heating and cooling durations. And actually, after a while, well, they came up with optimal regimes. They seem to be forming at least something. And when they analyzed, they got a non-negligible concentration of shorter peptides, of course, but that might be okay for our purposes. So they went further and scorched low-tech at this point a few different amino acids. And what seems to be happening, those different amino acids seems to be responding differently to heat. And what I mean by it, there seems to be different profile of formation and degradation as a function of time and temperature. And these results at this moment are simulated, but what Kuhan, Richin, and Masashi trying to do is to find out whether if we had some more creative heating schedules and we applied them to mixtures of amino acid, can we actually control or somewhat control the sequence of peptide formation? So incidentally, we're working on understanding better Fox's work. And so right now, I would like to switch gears a little bit and talk about projects in artificial chemistry that part of the MESI chemistry group have been working with. And so this is a very simple system. So what you have here is the solution with a large number of different components. And so this solution undergoes evaporative cycles, but not to a complete dryness. And precipitation of part of the components of this mixture are driven only by intermolecular interaction. We only get co-precipitation. And at this point, supernatant is containing something that hasn't been precipitated, is discarded, and fresh solution is added. And so what you do, you go lather, rinse, repeat until you achieve steady state. So that is somewhat built in into the model itself. Model is more specific at low temperature and less specific at high temperature. So what's happening here, you at the steady state and some given state, in red, your system remains messy. And if you go, when you go further at low temperature, system becomes sparse, which is interesting. And even more interesting here, they decided to test whether this system responds to any sort of perturbation, any sort of change in the environment. So what they did here is two arbitrary environment, and in like few steps, they linearly varied the parameter space of this state, trying to blend one state into another. And what they did here, they tried to follow concentration of certain components. And what we're finding here is that in some cases, certain components dominate one condition and certain other dominate another. So what we're trying to say, we can have this example of very idealized, but nevertheless adaptable system that can undergo rudimentary evolution. So what I'm hoping this our complex system group will do soon is with few tweaks to that model will help me understand actually my own results. This is my first experiment with Hyperbranch polyesters. So what I did in this experiment, I synthesized citric acid and glycerol polymer, and took me some trial and error to figure out that I want to do it with 1 part citric acid, 2 part glycerol, because when you do it 1-to-1, you get cross-linked polymer rather than Hyperbranch one. And here I was and I also, what I wanted to do is to see if the divalent cations, any salts, have any influence on the polymers I'm getting. So what's happened here, I synthesized this polymer under drying in neat. And when you analyze it by mass spec, so what you would expect, what would happen is you get a lot of glycerol-rich species. This follows the stoichiometry of the starting material. And interestingly enough, when I added calcium chloride, well, the system became more sparse, and there some of the peaks are given rise to species that are citric acid rich, glycerol poor. And what I trying to understand here, so what I'm thinking happening here is this, that citric acid chelates my calcium over here, and at any given time, some of the acid of the citric acid are blocked toward the esterification. So ultimately, you need more to include more of citric acid to get a polymer. And actually, I've been very good, so I'm not standing between you guys and your beer. So I want to conclude. So just to remind you, we're thinking origin of life is the transition between messy chemistry and well-orchestrated biochemical networks. So messy chemistry can give rise to a number of adaptable and possibly evolvable systems accessible through experiment or computationally. And one last thing, tarry materials might be a nightmare for synthetic and analytical chemists, but they actually might be very important in our search for origin of life. And just want to mention, this work has been funded by WPI, by EON, by Kokenhi, and my work with George Cody has been funded by Simons Foundation. Thank you for your attention.
right. Do you have any questions, Victor?
Sorry, so for Darwinian evolution in particular, for Darwinian selection, you need some sort of inheritance, some way of getting a somewhat reliable inheritance. Have you had any thoughts about this?
Well, Nathaniel certainly did, if he can get a microphone.
Well, so traditionally in evolutionary biology, we think of evolution as requiring reproduction with heritable variability and selection. The point here is that the first two seem to be pretty hard to achieve in chemistry, but the third one is really easy, right? I mean, if you just have, so for example, in that last model, there's no reproduction, but what you're doing is you're starting with a mess, a bunch of random things, and in the end, you're selecting the ones that are able to interact with each other in the right way to become precipitated in the dry phase. So so that's the key point there, and then the idea is that if selection can get you from a mess to a functional sparse system, then perhaps selection itself can be a help in getting you towards the types of systems where you're able to have replication and heredity. So we haven't done that yet, but that's where we're going with it.
Okay, I have one. You mentioned about Sidney Fox's work, which the bad you showed that he didn't show any kind of mechanism.
Mm-hmm.
How important is mechanism on a messy system? Because from my understanding, a mechanism is when you do clean chemistry, you can figure out the mechanism with various technique. In a messy system, how can one do it?
Well, and just if it's like mechanism, like we're studying in organic chemistry books, or it's not, but there need to be a reason. You need to show that this is not a fluke, right? There's need to be some chemical reasoning,
For
right?
the synthetic or for your messy chemistry?
For messy chemistry,
Okay.
right? I mean, as a chemist, I want to see a reason. I want to see that, it's not random, it's not just a fluke. I want to understand what's happening there. And I don't want like very specific mechanism of, shifting electrons here and there. Just kind of a set of plausible steps that can lead you from A to B.
But again, what you want and what you get is two different things, right?
No, that's true.
Yeah.
That's true.
So, we have any other questions? Sean, step.
Hey, thanks for the first formal Yokozuna lecture. And I was hoping you could talk a little bit more about the impact of the calcium ions in your polymerization.
Mm-hmm.
And more broadly, I started to wonder about different cation sizes. So for example, if you had a transition metal in there or something
certainly.
with a different ionic radius, what types of effects might you start expecting to see?
So yeah, this work published, and actually in this work I had series of different divalent cations and transition. There doesn't seem to be a difference. The results in every case in this particular system are the same. But this is the start, this is somewhat messy, and it needs a closer look.
Do you foresee structure coming out of this? Anything similar to peptides, or is it all just pure chemical systems that you're creating here from the messy chemistry?
From the messy chemistry, let's say structure, when I talked about hyperbranch polymers, right? And so
Sure.
Here I'm talking about, we know that most enzymes are globular. This system are also globular. So, I mean, there should be some generality to structure in that sense, right? Not necessarily in like sequencing and folding, but it's just kind of this general shape can be similar in that sense, I think. But, that's just some steps how you get from like this general shapes to the, shapes as they're expressed in current biology. That's a different question. I don't have an answer yet.
Okay, would you expect like a consistent morphology to come out of it, consistent shapes like beta sheets, or helices or something that could be universal between your messy systems?
Well,
Did you get
no,
some
you know what?
flexibility?
That's a very good question. No, I don't predict those. Just that there's going to be some general similarity. Maybe within those structures they would learn to support something like beta sheets and alpha helixes, maybe through deoxypeptide backbones. But, it's, how should I put it? Alpha helix and beta sheets are so unique, seem to be so unique to linear peptides. Yeah, it's going to be, in a large sense, it's going to be hard to replicate. Microphone?
Microphone.
Oh, there
Thank
it
you. Thank you. This is more of a comment. I thought that was fantastic.
Thank
and
you.
what I'm wondering is whether you're really, with the origin of life, talking about not just messy chemistry, but I've come to the conclusion it's a sloppy ecosystem. And that any attempt to have an origin of life based on some neat little single molecule is going to fail. That it was some kind of messy ecosystem, and life has always been sloppy and always complex, and that's the nature of life.
Well, thank you. That just seems to be necessary. So biology these days is quite messy. Am I right, Sean? And he's our resident biologist.
Just,
Victor, go
just to
ahead.
follow up on that one, how do you see the steps coming on? So, for example, when does a membrane come up? When does heredity come? So, which are the steps? I mean, if you've had any thoughts about this, I know
Yeah,
it's
I
not
don't have
big.
any thoughts about that. So actually, this system, I don't have a scenario here. So I'm here thinking about, for example, protoenzymes only. Yeah, it's hard. I mean, actually, I
You have to
haven't
clean it
thought
at some
yet.
point, right? So
I'll have to think at some point what comes first. At this point, I don't know, and I don't know.
you have to get from the messy one to a cleaner one.
Well,
clean
some
in a way, but not
control.
in
Yeah,
another.
in a way. You
Yeah.
need some better control, better
Exactly.
catalyst, better enzymes. So when membrane comes into compartmentalization and whether it's important to clean up messy chemistry, maybe it is because you can have a diffusion through a membrane, right? Yeah, membrane is exactly one way to clean up the mess. But yeah, so we don't have a particular scenario. In each case, and each selective pressure needs to be explored.
Koski.
Oh, thank you so much for the talk. So I'm kind of envisioning a transition from the messy chemistry to like a world where the translation systems start to like emerge. So I think the talk by Lauren Williams was like one of the functions of the ribosome is to actually make a linear polymer compared to these branching polymers. So if there's these like backup system running behind to actually do all the metabolic network, and you start to have these emerging ribosome, will there be inhibitory effect from these messy branch polymers against those rising, ribosome? Does it actually support, or would it kind of block the interaction between the co-symbiont of peptide and RNA? Sorry, it might be a little.
No, maybe yeah. I don't know. I don't have an answer. It's hard to know. It's hard to envision all the mechanism at this moment. So, what I understand, we had this conversation with Chris Adami once. So the reason that maybe life chose linear polymers, those are easier to encode for, easier to replicate. And we talk about branch polymers, and that he didn't see any way of, in that way it's going to be some sort of photography, how you would photograph a structure. So in that sense, these things actually might be inhibiting. But, what are the exact mechanisms or number of mechanisms, I don't know yet.
I guess we can do some experiment. For example, mixing these branch polymer with, for example, RNA strands and peptides and like everything together, and see actually how these interact. Do kind of selection. I mean, we'll talk back later.
Mess to the rescue, yes.
I have a question again. Me. Yeah. Messy chemistry, systems biology, why the words are changing all the time?
Oh, yeah, that's a tough question. How should I say? So systems chemistry, system biology means so many things to so many different people. And we wanted a different term. And so some of, as you talk to them, they don't really like the word messy. It's really not precise. But anyone of like prebiotic chemist who worked with messy system with prebiotic chemistry, they understand what qualitatively messy means right away. So I think that was my thinking when we decided to call our chemistry messy.
Do you have any last questions? This is the last talk of the day. Any biting questions? Jim Clips.
Nice talk. I wanted to ask your opinion. So earlier in the week, we heard this kind of notion that life built itself up from simplicity into complexity. And this seems like completely the opposite notion. Is there some way to reconcile those two concepts?
Well, it just all depends how you define simplicity and complexity, I think. So in what sense? I just, if you're just talking about chemistry, I can't envision like very clean chemistry happening without enzymes. On the other hand, when you just like different kind of processes, different kind of chemistry happening here, that's actually a limited amount. Chemistry is simple, structures are complex. And when you're going to biology, I think that, the chemical space is vast, but, structures are relatively not simple, but similar, repetitive. Does that answer your question?
Well, yeah, there's a lot of ways to answer it, I guess.
Yeah, I know.
Yeah.
All right then. Thank you. Let's give a hand to Irena.
Thank you.